So let's do the notes on this section, which is comparing size and order of magnitude. So that's what order of magnitude is. It's a way of comparing size. Differences in size can be expressed as an order of magnitude. This means how much bigger or how much smaller something is compared to something else. When objects are approximately the same size, they are of the same order of magnitude. If an object is 10 times bigger than another, it is one order of magnitude bigger. If an object is 100 times bigger than another, it is 2 orders of magnitude bigger. The easy way to figure that out is actually just to count the number of zeros. 100 can also be expressed as 10 to the power 2, which is 10 squared, and it's the power, that number, is the order of magnitude. You may have to look at putting some cells or living organisms or a mixture into a size order. So here's an example from smallest to biggest. Virus, which is smaller than bacteria, which is smaller than an animal cell, which is smaller than a fly. And then if we look at the table, there's a comparison there of the order of magnitude expressed in standard form. So one order of magnitude means 10 times bigger, times 10, and the standard form version is 10 to the 1. So that order of magnitude is the power. So 2 orders of magnitude also means times 100, so 100 times bigger, which in standard form, or the version which involves the power, would be 10 to the power 2, or 10 squared. So that 2 is the order of magnitude. And then again, 3 orders of magnitude is 1,000 times bigger, which is 10 to the 3. And 4 orders of magnitude is 10,000 times bigger, which is 10 to the power 4. Quick reminder of units here, because they're going to be important with the calculations. So a centimetre is worth 10 millimetres. A millimetre is obviously, we're just looking at converting to millimetres, so that is 1, but we're just putting it in the order so you can see the order. The micrometre, that little symbol, is 0.001 millimetres, and then a nanometre is even smaller, 0.00001 millimetres. But the conversion chart is at the top, which that makes things quite a bit easier to convert between the different units, and you just follow the units around. So if you were starting with a number in micrometers and wanted to get it into millimeters, you just follow the arrow and use the function there. So that would be divide by 1,000. So keep that little conversion chart handy. That really does help, especially when you're just getting used to the calculations. So here's an example question. If a virus is 100 nanometers and a bacteria is 1 micrometer, what is the difference in order of magnitude? So you need to be working with numbers that are in the same units first of all. It doesn't really matter which way round you do the conversion, but I would go for getting both into nanometers. That looks like it would be a little bit easier for me. So the 1 micrometer is equal to 1,000 nanometers, and we've also got the virus is already in nanometers, so we don't need to convert that. So what that tells us is the bacteria is 10 times as big as the virus. So we've just done 1,000 divided by 100 there to get times 10. So 10 is 1 order of magnitude, or in standard form 10 to the 1. So that's the answer. Okay, so that is the notes done there. And then we've got the master it, so do pause the video here and have a go at these questions. And then when you're ready, we'll go through the model answers. So putting those collection of things into an order. Smallest is virus, then bacteria, then animal cell, then fly. And looking at the questions, what is the difference in order of magnitude between a leaf cell of 100 micrometers and a virus of 100 nanometers? So the leaf cell is 100 micrometers, which we can convert. So use the chart. If we start at micrometers here, we want to get into nanometers to get both of those numbers into nanometers. So you follow the arrow round and we need to multiply by 1,000. So 100 times 1,000 equals 100,000 nanometers. We need to find how many times bigger by leaf cell size divided by virus size. So 100,000 divided by 100 equals 1,000 times bigger. And 1,000 is 10 to the power 3. And there are 3 zeros, so there are 3 orders of magnitude of difference. Number 2, what is the difference in order of magnitude between an ant of length 1 millimeter and a cheek cell of length 100 micrometers? So let's get those both into the same units. The ant length is 1 millimeter. That's equal to 1,000 micrometers. We're finding how many times bigger. So we do that by taking the ant length divided by the cheek cell length. So 1,000 divided by 100 equals 10, or 10 to the 1. Therefore, there's 1 order of magnitude of difference. So you can either count the number of zeros or have a look at the power, and then you're done. Okay, so there's some quick quiz questions all related to that sort of stuff. So pause and have a go at those. Either work on your own or with a partner, but keep doing it on repeat until you get that perfected. And then you've got the answers as well to double check. So the next set of notes is on microscopy. There is a required practical microscope, so all the labelling and the practical skills will be covered separately, but we'll look at the theory here. So let's do the notes. So microscopy. What are the 2 types of microscope and how did they develop over time? So light microscopes use light and lenses to magnify cells so we can see them. With a light microscope, we can also see some organelles such as a nucleus. A light microscope has fairly low magnification at 1,500 times and a resolution at 200 nanometers. Electron microscopes were developed later. In 1931, they use electrons to form an image. They have the advantage of a higher magnification of 500,000 times and a resolution of 0.1 nanometers. Therefore, electron microscopes help us see more details such as more organelles. What does magnification and resolution mean? Magnification means how much larger the image is compared to the real size. Resolution, or resolving power, you might see it referred to as, means the ability for the microscope to distinguish 2 close together images as being separate. So you know, if you've looked at onion cells under the microscope, it's the light microscope that we usually have in the classroom, and we won't be able to see the cell wall and the cell membrane as 2 separate images, and that's because of the low resolution of the light microscope. There's a little formula we might need to use at the top and possibly need to rearrange it. So magnification equals image size divided by actual size. Rearranging can be done by algebra, or some people like to use this little triangle method. So you might have to get the formula into the triangle. So here we would have image size divided by actual, so image would go at the top, divided by actual, so that would go underneath, and then the remaining spot is for magnification. So if you look at the way this triangle is set up, it's quite helpful to be able to go from formula to triangle and then triangle to formula. Standard form is quite a crucial skill for this topic, so here's a little reminder that the standard form version of 0.0025 is equal to 2.5 times 10 to the minus 3. So this is just a little reminder of this skill. It is covered in maths, it's covered obviously in biology, and there will be a bit of practice coming up on that as well. So the number of places the decimal point moves becomes the power, and if the decimal point moves right, then the power is negative. So here's another example, 0.017 equals 1.7 times 10 to the minus 2. So that first number has to be between 1 and 10, but less than 10. A worked example, so this is the sort of question that will come up. A cell that is 17 micrometers in diameter appears to be 34 millimeters in diameter when viewed through a microscope. Calculate the magnification. Write the equation, then write out the values, then check the units in case you need to do a conversion. So I would always suggest writing out these steps, because sometimes if the final answer is not correct, then there are marks that can be gained for the steps. So magnification equals image size divided by actual size. The magnification, we don't know that yet. The actual size equals 17 micrometers, and the image size is 34 millimeters, so we need to be working with both sizes in the same unit. So let's convert that millimeters into micrometers. So use that conversion chart from before and multiply by 1000 to give you 34,000. So then we are putting the numbers into the equation. Magnification equals 34,000 divided by 17. So the magnification is 2,000 times. Okay, so that's how many times bigger the cell appeared compared to what it actually is. So that's the notes completed. There's a little bit of practice here on standard form and conversions. I have included the little conversion chart at the bottom there, so you can use that for a bit of support if needed. So pause the video and have a go at those. Let's quickly read through the answers for you. So number 1, 17,000 in standard form is 1.7 times 10 to the 4. 0.006 equals 6 times 10 to the minus 3. 250 equals 2.5 times 10 to the 2. 0.00008 equals 8 times 10 to the minus 6. 740,000 equals 7.4 times 10 to the 5. Then the other way, converting standard form into a number. Number 1, 4.5 times 10 to the 3 equals 4,500. Number 2, 1.8 times 10 to the power 5 equals 180,000. Number 3, 2.9 times 10 to the minus 2 equals 0.029. 5.6 times 10 to the power 8 equals 560 million. 7.4 times 10 to the power 2 equals 740. Conversions, right, I'll work this through with the chart. So if a cell measures 2000 nanometers, so that's here, How many micrometers is this? So we are going from nanometers to micrometers. That's divided by 1,000. That gives us the answer of 2 micrometers. If a cell measures 5,000 nanometers, how many micrometers is this? So we start at nanometers to micrometers again. So again, that's divided by 1,000, and the answer is 5. If a cell measures 70 micrometers, how many nanometers is this? So micro to nano, so it's times by 1,000, so that's 70,000. If a cell measures 45 micrometers, how many nanometers is this? Again, times by 1,000, which is 45,000. Number 5, a bacterial cell is 1/10 the width of a eukaryotic cell. The eukaryotic cell is 2 micrometers wide. Calculate the width of the bacterial cell. So first of all, you've got to divide the eukaryotic cell by 10, which is 0.2. And so that's the width, but in nanometers, so we would go from micrometers, 10 nanometers here, times it by 1,000, and that is also equal to 200 nanometers. So either of those answers there would be absolutely fine. Then if you have to give that answer in nanometers using standard form, then that will be 2 times 10 to the power 2. So that's going from the nanometer into standard form. So there's a little bit more mastery here, and that's to practice some exam style questions on microscopes. So pause the video now and have a go at those. So I'll have a look at the answers with you, but of course if all you need to do is quickly check them, you can then get on to the next section. 1. Calculate the magnification of a cell that's 12 micrometers wide and appears 3 centimeters wide under the microscope. So write out your formula. Magnification equals image size divided by actual size, where the image size is 3 centimeters, or that into micrometers. First times it by 10 to get it to millimeters, and then times it by 1,000 to get it into micrometers, which gets us to 30,000 micrometers. And the actual size is 12 micrometers. So the magnification equals 30,000 divided by 12, which is 2,500 times. Number 2, calculate the magnification of a cell that is 25 micrometers wide and appears 5 centimeters wide under the microscope. So again, write the equation. Magnification equals image size divided by actual size. Image size equals 5 centimeters. So into micrometers, you need to first multiply by 10 to get it into millimeters, and then by 1,000 to get it into micrometers. So it's 50,000. The actual size is 25 micrometers. So then do the calculation. 50,000 divided by 25, which is 2,000. Number 3, an onion cell is viewed under a microscope with 100 times magnification. The image of the cell is 7.5 millimeters wide. What is the actual width of the onion cell? Give the answer in micrometers. Okay, so we're finding the actual size this time. So use the formula triangle if you need to. So because we're finding actual size, we cover that up with our thumb and see what we're left with. So that means we have to do image size divided by magnification, which I've written there for us. And then the image size is 7.5 millimeters, which is 7,500 micrometers, because that's just to multiply by 1,000. So then the actual size is 7,500 divided by 100, which is 75 micrometers. Number 4, a muscle cell is viewed under a microscope with 200 times magnification. The image of the cell is 5 millimeters wide. What is the actual width of the muscle cell? Give the answer in micrometers. So again, we've got another actual size to calculate. So that again is image size divided by magnification. We've got the magnification, that's 200. We've got the image size, which is 5 millimeters. But let's convert that to micrometers because that's the unit we're required to give the answer to. So that's a millimeter to micrometer times by 1,000. So that's 5,000, and so the calculation is 5,000 divided by 200, which is 25, and that's in micrometers. Number 5, what is the size of the image formed when a nucleus of actual size of 6 micrometers is viewed under a magnification of 8,000 times? Give your answer in millimeters. So the image size we want this time, so we go to the triangle, cover up the image size, what we're left with, and we're left with actual size times by the magnification. So that's written as the first step. The magnification is 8,000 and the actual size is 6 micrometers. So the calculation is 8,000 times by 6, which is 48,000 micrometers. Now we've got to convert that back into millimeters at the end. So micrometers to millimeters divided by 1,000 is 48 millimeters. Our next section, let's do the notes for estimations. So again, this is another one of those math sort of skills, and it's found its way into our specification, so we have to practice it here. So estimations. Estimations can be used to work out a rough answer. Using estimations in cell biology, you can estimate the diameter or area of organelles that are too small to measure. If you know a cell is 30 micrometers long and 10 micrometers wide, you can estimate the area of the nucleus. Work out the area of the cell using area equals length times width. Area equals 30 times 10, which equals 300 micrometers squared. Work out approximately how many nuclei would fit inside the cell. So I've got an approximation down here. So we thought that 6 could fit along lengthways, and then you could get 3 rows of those, so that takes us to about 18. So then divide the area of the cell by the number of nuclei. So 300 divided by 18 equals 16.7 micrometers squared. To estimate the diameter of the nucleus, work out approximately how many nuclei would fit across the length. So like we said here before, there's 6 that fits along the length. So we divide the length of the cell by the number of nuclei. So 30 divided by 6 equals 5 micrometer. So here's a question to practice. Pause and take as long as you need. So a cell has a length of 40 micrometers and a width of 15 micrometers. Estimate the area and diameter of the nucleus and round your answer to the nearest whole number. So work out the area of the cell. So that's area equals length times width. 40 times 15 equals 600 micrometers squared. Work out approximately how many nuclei would fit inside the cell. So we thought about 1, 2, 3, 4, 5, 6, 7 in this case across and 4 down. So in total that would be 28. If all these nuclei are all filled in here, there'd be 28 of them. So we're dividing that whole area of 600 by the number of nuclei, so by 28, which gives us 21 micrometers. To estimate the diameter of the nucleus, work out approximately how many nuclei would fit across the length. This is about 7 we estimated. And then divide the length of the cell, which was 40, divided by the number of nuclei, which is 7. And that gives us 6 micrometers. So answers are typically rounded when carrying out estimations, but check to see what the question asks for. If it asks for give your answer to 1 decimal place, then do that. But if it doesn't ask for anything, then just go for rounding in this case. There's another quick quiz for you to have a go at. So it's always quite fun to get together with somebody at this point and then just do it on repeat. One of you asks the questions while the other answers, swap over and keep doing it and doing it until you get it perfect. There's the answers. Now let's discuss the required practical using a light microscope to observe, draw and label a selection of plant and animal cells. So how to prepare a slide. You obtain a new glass slide and add a drop of water in the middle. Place your specimen into the water on the slide, such as a thin layer of onion tissue. Add a suitable stain, for example, add iodine to the onion tissue so you can see parts of the cell. Cover the specimen with the cover slip by placing one side of the cover slip onto the slide, then slowly lower until flat, and this prevents air bubbles getting trapped. The prepared slide goes on to the microscope stage to view the cells. And how to view the cells with a microscope. So place the prepared slide onto the microscope stage. Set the lowest power objective lens to its lowest position closest to the slide using the coarse adjustment knob. Look down the eyepiece and turn the coarse adjustment knob to bring the cells into focus. Use the fine adjustment knob to get a clearer image. Work out the magnification by multiplying the objective magnification by the eyepiece magnification. And then we have to be able to know how to draw the cells. So use a sharpened pencil to draw the cells. Use clear lines, do not use shading. Draw the outline of the cells, add any features that you can see inside of the cells, and to proportion as best as possible anyway. Give the drawing a title, label the parts of the cells, and add the magnification that the cells were observed at. So pause the video here and have a go at the questions using the changes of colours if you need the support. And then let's go through those answers. So describe how to prepare a slide. Take a glass slide and add a drop of water. Place specimen into the water on the slide, such as a thin layer of onion tissue. Add a stain, such as iodine, to the onion tissue so it can be seen. Cover with a cover slip, slowly at a tilt so that air bubbles do not get trapped. Place onto the microscope stage to view the cells. So if there's anything to add in there, change the colour again, otherwise give a tick for each point. Describe how to view the cells with a microscope. Place the prepared slide onto the microscope stage. Set the lowest power objective lens to its lowest position, closest to the slide using the coarse adjustment knob. Look down the eyepiece and turn the coarse adjustment knob to bring the cells into focus. Turn fine adjustment knob to get a clearer image, and work out the magnification by multiplying the objective magnification by the eyepiece magnification. Describe how to draw the cells. So use a sharp pencil to draw the outline of the cells and features to proportion. Do not shade, use clear lines. Add title and labels. Add the magnification that the cells were observed at. And then for drawing the onion cells, they should look a bit like this. So no overlapping, clear kind of lines. You can kind of see a thickened outside layer because the cell can't, with the light microscope, we can't distinguish between the cell wall and the cell membrane, so it kind of looks like a thick outside layer. So it's okay to draw it as a thick outside layer, but you can't label both because you can't distinguish both. So for the plant cells that we view under the light microscope, we usually label the cell wall because we can see an outermost layer, but we can't distinguish the cell membrane separately due to the low resolution. But we can often see a nucleus or several nuclei, so we can get one labelled. And then the stuff that's all inside is the cytoplasm, so that can be labelled as well. Then with your objective lens, if you were looking at a ×10 magnification, together with the eyepiece lens, which is typically a ×10 multiplication there. So you multiply the two together, in which case this would be a ×100. But if you had your objective lens on ×4, then together with the eyepiece lens of ×10, that would overall be ×40. So it depends on your microscope. This next required practical does not apply to the combined science, so it applies to Edexcel and AQA as a single science in biology. So this practical is to investigate the effects of antibiotics or antiseptics on bacterial growth using agar plates and measuring zones of inhibition. So you have to be aware of how to grow the supply of bacteria that's going to be tested, and then how to set up a lawn of bacteria using an aseptic technique. So we'll go through those, and they are equally important. You may not have to actually carry those parts out practically, it depends on your situation, but you'll certainly carry out the investigation step by step below. So we'll go through the growing bacteria first. So bacteria can be grown in a solution of carbohydrates, proteins, vitamins and minerals, and this is called a nutrient broth solution. Bacteria can also be grown on a nutrient-rich jelly in a petri dish. This is called agar jelly or agar plate. Antibiotics and aseptics can kill bacteria if they're not resistant strains, so that links in with the antibiotic resistance in another topic. The area around the antibiotic or antiseptic where bacteria die is called the inhibition zone. So that's a really important term. The aseptic technique that means working in a way to prevent contamination. So prevents other species of bacteria kind of getting in to your investigation. You must use sterilized agar jelly and sterile petri dishes to avoid contamination. You will transfer the bacteria to the growing medium with an inoculating loop, which must first be passed through a hot Bunsen flame to sterilize it. Or use a sterile dropping pipette and spreader, and these tools create an even lawn of bacteria once they've grown. A bit like a grassy lawn, which is why it's called a lawn. Then investigating the effect of antibiotics or antiseptics on these bacteria that have been prepared and have grown. So number one is to soak paper discs in a variety of antiseptics or antibiotics, for example, bleach, hand sanitizer, toilet cleaner, kitchen cleaner, and also include sterile water. So that will be the control experiment. Then, number two, place a soaked disc, keeping record of which is which, from each solution onto a prepared agar plate with a bacterial lawn. The water-soaked disc acts as a control to ensure it is not the presence of the disc that kills the bacteria. So we want to make sure it's the antibiotic or antiseptic that does that and not just the disc. That's why we soaked one in the water. 3. The lid is taped lightly on. In 4 places is ideal, like in kind of the corners, if they were to have corners like you would on a square. So in other words, not all the way around, just in 4 spaced places. This prevents contamination from the microorganisms in the air, but does not prevent oxygen, which is needed for respiration. So they don't kind of get killed off due to lack of oxygen. The agar plates are stored upside down to stop condensation falling on them, and are stored at 25 degrees C for 48 hours. So now have a go at the questions to properly master it, and pause the video to give yourself enough time to have a good go at that. I might just point out a couple of things for you. So the diagram, you've got the big circle is showing the agar plate with the lawn of bacteria spread. That's kind of that light bluey sort of colour. And the paper discs that were soaked in the antibiotic or antiseptic are that dark blue colour there. And I've got labelled the inhibition zone. So as we said, that was the area that gets killed off by the bacteria. So if you pause the video here and have a good go at these questions, change colour if you do need support, and we'll go through the model answers in a moment. Okay, so looking at the model answers, so you improve it colour for anything that you didn't have in there already. So the most effective antibiotic or antiseptic will create the biggest inhibition zone around the disc. The diameters of the inhibition zones must be measured. These are divided by 2 to find the radius. So a little bit of maths skills here. And to calculate the area of inhibition zone, the following formula is used. Area equals pi r squared. So the pi button on a calculator is pressed. So that's approximately 3.14, but there are lots of other numbers after that as well. So just always use the pi button. Then it's multiplied by the radius squared. So it's the radius part that's squared only. So describing the aseptic techniques that can be used when growing organisms. So it's using sterilized agar jelly. and sterile petri dishes to kill the unwanted bacteria. Transfer the bacteria to the growing medium with an inoculating loop, which must first be passed through a hot Bunsen flame to sterilise it. Always hold the lid of the petri dish at an angle to stop bacteria from the air falling in, and tape the lid on lightly. Then to the diagram, which is not to scale. Number 1 is the diameter that's measured across the middle, so that goes right the way from one side of the circle of the inhibition zone. It might not always be a perfect circle, that's just for the textbooks, but it usually is approximately, but you get some weird sort of shapes along with it as well. So not an exact circle, but approximately. The diameter is divided by 2 to find the radius. 3, the area is calculated using pi r squared. 4, the most effective antiseptic has the biggest inhibition zone. So calculate inhibition zone A and B. The diameter of A is 10 mm and B is 14 mm. 2, the radius of A is 5 mm and B is 7 mm. So the two numbers in number 1 were just divided by 2, of course. 3, area A, so it's pi times 5 squared, and B is pi times 7 squared. So number 4, the area of inhibition zone A is 79 millimeters squared, and the area of inhibition zone B is 154 millimeters squared. So that was number 3 has just been worked out in the calculator to give those numbers. And then 5, the most effective antiseptic is B because it's got that biggest inhibition zone. Practice naming your variables. It's always an important skill throughout all of the practicals. So in this investigation, name the independent, dependent and control variables. Press pause to give yourself enough time to complete. So let's go through the variables for the practicals. So the independent variable was the one that we changed. That was the type of antiseptic or antibiotic. The dependent variable was the one that we measured. So that was the diameter of the zone of inhibition around each paper disc. Then we controlled as many of the other variables as possible. So we kept these things the same paper discs and the same bacteria were tested. So well done everyone, that's it for this tutorial and see you next time.